This is Christopher Sernaque hosting episode 2 of season 4 of the Current Topics in Science podcast. This podcast will address breaking scientific news in light of the origins debate and host interviews with scientists. This podcast is available on the following platforms: iTunes, Stitcher, Google Podcasts, Spotify, and iHeartRadio. Video recordings of the podcast will be uploaded to YouTube. Enjoy the podcast. Today on Current Topics in Science, we have the honor of hosting David Thomas. Currently an author for Creation Ministries International, David Thomas, using his pen name, is writing cutting-edge research on the design of molecular motors. David Thomas is currently studying biological sciences, cell biology, and genetics at a secular university. He currently has over 60 different articles and research papers planned out. And in this interview, David and I will be going over evolutionary models and molecular machines, and in our next interview on the other podcast, Pilot's Interview, we're going to be discussing David's advice for fellow Christian university students at secular universities. So David has several book ideas, research projects that are upcoming, and you can read his first article on the E. coli's electric motor, which he calls a marvel of design, in the description below. And so now, without further ado, good afternoon, David. How was your day, and how are you doing?
I'm quite tired, but doing well.
It's fantastic to have you on, David. And since this is Current Topics in Science, we're going to quickly look at this week's current topic. Inside Science reports a bacterial enzyme that copies DNA might make more mistakes in zero gravity, study finds. According to Sylvian's article, the paper raises the possibility that some enzymes work differently in space compared to on Earth. It gives us an idea that enzymes, like polymerases, or others that are involved in maintaining the integrity of our DNA, may be influenced by spaceflight, said Susan Bailey, a radiation cancer biologist at Colorado State University in Fort Collins, who has studied DNA damage caused by space radiation and did not contribute to the new paper. So the scientists studying this effect, Rosenstein, took a trip aboard the Vomit Comet, which is a type of plane that achieves zero gravity by performing a series of ascents and dives in quick succession. Inside Science reports that Rosenstein studies DNA polymerases, a class of enzyme that copy DNA and repair damage. He conducted an experiment aboard the Vomit Comet where he used a clicker to prompt robotic instruments to inject a polymerase found in the E. coli into a mixture holding a synthetic single-stranded DNA. As the plane left zero gravity, he injected another mixture to stop the reaction. So this was the conclusion of his research, as summarized by Inside Science. It was that an enzyme in the bacterium E. coli made more errors copying synthetic DNA when exposed to zero gravity than the same enzyme did in normal gravity. David, you've written an article on the electric motor of the E. coli. What can you tell us about this organism, and what do you think about this current topic?
E. coli is a species of bacteria that is commonly used for research because it's quite easy to culture. But this study is quite interesting, the finding of the study, because gravity is normally ignored when discussing molecular machines, because at their scale, then the random jostling of water, which is referred to as Brownian motion, has a much stronger effect than gravity. So the smaller you get, the more Brownian motion has an effect compared to gravity. And so by the time you get to a single molecular machine, then the effect of Brownian motion is just so much stronger than gravity that gravity is normally just ignored. But what the study shows is maybe we shouldn't just ignore gravity, because it seems that it can still have an effect on the function. The study also provides evidence that molecular machines are designed to work optimally on Earth with gravity, because it seems that they don't work as well without gravity. And related to this topic, there are eukaryotic cells, which are cells with a nucleus, that sense gravity using a gravity sensor. They have a heavy weight which sinks downwards and either pulls on sensors or pushes on sensors, which the cell then uses to respond to gravity. However, there are microorganisms which swim upwards away from gravity to stop them sinking in water, but they don't seem to have any heavy weight. And so it's not entirely clear how they can detect gravity, because any gravity sensor without a heavy weight would be far more strongly affected by the jostling of Brownian motion than by gravity. So I'm sure the study will lead to some interesting findings.
Well, thank you for commenting on this new research, David. It's pretty incredible when you think about it, because it shows how finely tuned, as you said, the Earth is for life. Speaking of which, in your own research on the flagellum, you've unlocked some startling insights. Your article had several headings covering the motor parts, gear mechanisms, motor construction, and the genetic construction program. So, David, can you tell us about each of these different parts or components of the flagellum on the E. coli?
The flagellum motors are just amazing. Like the similarities between flagellum motors and human design motors are just simply astonishing. There's so many parts in flagellum motors that are so similar in their function to human designed motors, including gears, but they've also got rotors and axles and drive shafts and bearings and brakes and clutches and structural scaffolds and oil-like lubricant and hinges and universal joints and other joint types and adapter rings and switches and stators and capacitors and mechanical sensors. And like they're just so similar to the motors that we design. Now, the most studied flagellar motors are those in the species E. coli and also Salmonella. But the motors of most species are far more complex than the motors in E. coli, and they have many additional parts. But we don't yet know the function of those additional parts, so we don't know the function of most of those additional parts. So I expect that as more research is done, then we will uncover more similarities between human designed motors and the motors that God designed in bacteria. There could, like, based on genetic evidence, then there could be thousands or even millions of different types of flagellar motor designs, which is just amazing, like having so many different designs of these molecular motors in bacteria. Some of these designs seem to be optimized for speed, while others seem to be optimized for power. One of the fastest flagellar motors has been recorded spinning at 102,000 RPM, which is much faster than a Formula One racing car engine. Now, talking about the gears, essentially the gearbox of the flagellar motor contains a large central gear, which is surrounded by and driven by multiple smaller powered gears. And the powered gears are powered by an electric voltage and chemical potential across the inner membrane of the cell. And so the cell is surrounded by a membrane, and there's a voltage across that which powers the powered gears, which in turn rotates the central gear to power the entire motor. And so, like, they literally are electric motors. Now, one of the amazing discoveries recently is all the different types of gear mechanisms that flagellar motors have. So, for quite a while, we've known that flagellar motors can rotate in forward and reverse gear, but we didn't really know how they could do that. But now we know that how they do it is in forward gear, the small powered gears contact the outside of the large central gear, but to switch to reverse, then the central gear expands so that then the powered gears contact the inside of it. And because the powered gears always rotate in the same direction, then that changes the direction of rotation of the central gear. But there's also other gear mechanisms. There's one to change power output. So if the cell swims into thicker fluid, then it's harder to rotate the propellers. And so the motors automatically detect this with mechanical sensors and automatically engage more powered gears with the central gear, which increases the rotational force produced by the motor. And then again, if it swims into thinner fluid, it's easier to rotate the propellers, and so less powered gears are engaged. And so what this means is that the motors only ever use as much energy as they need to, which makes them very energy efficient. Some species, the electric voltage is made by protons, whereas a difference in proton concentration across the membrane, whereas in other species, the motors are powered by a difference in sodium across the membrane. But there's still other species which have hybrid motors, which use both fuel types. And in these species, then these species have another gear mechanism where some powered gears use protons and other powered gears use sodium ions, and the motors can change the ratio of the powered gears, of each type of powered gear in the motor, to change the ratio of fuel type that is used in response to how available the different fuel types are. So again, you've got this like high efficiency with like changing which fuel the motors are using depending on how available they are. You also get species that have gear mechanisms to change the speed of the motor. It seems like it's not entirely known yet why they need to change the speed of the motor, but I'm sure more research will find that out. But there is also, I mentioned earlier that flagellar motors have clutches and brakes, and these are used essentially to slow the motors down when forming it's called a biofilm, which is a community of bacteria. And when they're in these communities, then they're not moving around, and so they've got the species have these systems which detect that they are coming near surface and going to form a community, and so they automatically engage clutches or brakes to slow down the motor to get it ready for attachment to the surface, and then the cells can be in the community. Now, you also mentioned in my article, I had a section discussing the construction of flagellar motors. The construction is just so amazing and just so complicated because every single bacterial flagellum is made of thousands of individual proteins, which each have to be assembled into several different parts, like each of the parts of the motor and the universal joint and propeller. And all of these proteins have to be manufactured in the cell at the right time in the right quantities, and then transported to the assembly site at the right time, and then assembled into the right location to each form each of the parts that they're meant to form. And each of those parts has to be made to the right size with the right diameter and the right length. And this is just a huge engineering challenge to be able to design something that could get all of those things correct would be quite a challenge. Last week, I was talking with an engineer who was talking to me about his work writing out the assembly instructions for aircraft jet engines. And I then talked to him about the assembly requirements for assembling flagellar motors. And because this is his area of work, like this is what he does for a job, writing out these instructions, then he was understandably quite amazed by all this. And as he thought about, from an engineering perspective, thinking of the systems and software that must be required to control the whole assembly of a bacterial flagellum. And he was correct that the construction is controlled by systems and software, much like computer programs and software that humans design. And the entire construction process, from the production of each of the proteins to transporting each of them to the assembly sites to controlling the assembly of each of the proteins into each of the parts, is all controlled and coordinated by program systems. The construction process has multiple different systems which work together and communicate with one another in a coordinated fashion to control every single step of the construction to make sure everything is done correctly. Because if it's not done correctly, then the flagellum just won't work properly as a motorized propeller. One of these systems is called the flagellar gene regulatory network, which I talked about in my article on E. coli's motor. This system controls the timing and logistics of producing all of the proteins required for the flagellum assembly. When you think about the fact that there are over 20,000 proteins which must be produced, with different types of proteins needing to be produced at different times and in different quantities, then that's a huge logistical challenge. And so the flagellar gene regulatory network controls the logistics of all this to make sure that everything is made in the right quantities at the right time. And I've put a diagram of this network in my article, and you'll see in the diagram it functions so much like a computer program with circuitry and feed forward and feedback loops, input and output signals, and also logic gate functions, which are used in man-made computers. It solves all the computations in a very similar way to man-made computers, using all of these circuitry and logic gate functions. And so the diagram I've got in my article is a simplified diagram. There's stuff I haven't included, because if I included the entire network with all the connections and stuff, then it would just be way too complicated. So the diagram may look complicated in the article, but that's only part of the system. There's also another system controlling the construction, which controls when each of the proteins gets exported to the assembly site to be assembled. This system controls two different types of molecular machines, which work in tandem to export different types of proteins into different places where they need to be assembled. So one of these machines is located inside of the flagellum itself, and that's called the export apparatus. And then you've got another type of machine called the secretion pathway, which is not located in the flagellum, but still exports proteins. This system also controls the length of the universal joint. In cells with multiple flagella, then the universal joint has to be made to the right length, because if it's too long or too short, then the cell can't swim properly because the propellers interfere with each other. And so the universal joint has to be made to the right length. And so the export apparatus literally uses a molecular ruler, which is it's a long protein, which is like a string, and it uses that to physically measure the length of the universal joint as it's being made. And then once it reaches the right length, then it stops the universal joint from being made, which makes sure that it's made to the right length. There's also another system which controls the length of the drive shaft, and it controls that based on the distance between the inner membrane and the outer membrane of the cell. So cells with two membranes surrounding them, then the drive shaft will grow until it reaches the outer membrane, which is the distance it has to travel, and then the drive shaft will stop getting built. And there's a system to control that. We're not entirely sure how the system works yet. There's also another system which controls which proteins attach to which other proteins, how strongly they attach, and what angle they attach to each other on. In flagellar motors, there's lots of parts which form a ring shape or a tube shape. And for these structures, then the angle that each protein binds to one another on in the ring controls the diameter of the ring. Because if they bind to each other on a really tight angle, then you'll get a small diameter ring with only a few proteins around the ring. But if you get a really wide angle, then you get lots of proteins forming the ring, which then changes the diameter of the ring. And so the system also controls the diameter of each of the ring and tube structures in the flagellar motors to make sure they're all made correctly. So you can see there's all these different systems controlling the whole construction process to make sure everything's done correctly, and all these systems work together just like man-made systems.
That is truly awesome research, David. That's incredible. Now, you've written a soon to be published article about this metal compass needle that some bacteria manufacture and use for navigation. So, while you did include an illustration and description in the article on E. coli motor about this navigational system, can you tell us more about the navigation systems of bacteria?
Well, once again, the navigation systems of bacteria, I've talked about the construction systems, controlling the construction, but with the navigation systems, then again, they function just like man-made systems. And with bacteria, they don't just swim around aimlessly, just as if they're lost. They swim around very deliberately, trying to get to from where they currently are, trying to get to where they want to be. And to do that, they have to have a navigation system which can process lots of signals from sensors and steer the cell in the right direction so that it gets to where it wants to be. So I'll briefly discuss the basics of how these systems work and how they solve computations, and then I'll discuss the compass needles and what they're used for. Bacteria like E. coli swim around in a three-dimensional zigzag. The cell propels forward in one direction, which is called a run, and then it tumbles to reorient in a new direction, and then it propels forward again in the new direction. And so they swim in this three-dimensional zigzag with runs where they propel forward and then tumbles where they turn. Now, if a cell is swimming in a favorable direction, the direction it wants to go, then it swims for longer in that direction than if it's swimming in a non-favorable direction, which then results in the zigzag being biased towards the direction that it wants to go to. So the navigation system calculates how long a run should be or how long it should travel in a particular direction based on the measured changes in chemical concentrations. So if the concentration of nutrients is increasing and the concentration of toxins is decreasing, then that's a favorable direction. And so the navigation system will calculate that the cell should go for quite a long way in that direction. But if nutrients are going down and toxins are increasing, then the navigation system will quickly stop the cell from going in that direction and will turn the cell, and then it will try a new direction. But how does the navigation system perform all these necessary calculations? The navigation systems in bacteria are made of thousands of core signaling units. Each core signaling unit is made of 18 proteins, and thousands of core signaling units assembled together into large arrays called signaling arrays. The signaling array functions as the computer of the navigation system, which solves all of the calculations. The signaling array receives numerous input signals from sensors that measure the current concentration of different chemicals. So that it could be good chemicals like nutrients or bad chemicals like toxins. The signaling array receives numerous input signals from sensors that measure the current concentration of different chemicals. So they could be good chemicals like nutrients or bad chemicals like toxins. The signaling array then compares the current concentration measurements of these different chemicals to the measurements performed a few seconds prior. The sensor array can then calculate the concentration difference between the current concentration measurement and the past concentration measurement to see if the concentrations are going up or down. So the navigation system has to have short-term memory to be able to compare the concentrations over time. And so they've got a short-term memory feedback loop so that it can make these comparisons. The sensor array then integrates all of these calculations into a single output signal, which gets relayed to the motor and determines how long a run will be or how long it will go in that particular direction. The signals get integrated because if the concentration of nutrients is going up, but the concentration of toxins is also going up and going up faster, then the cell doesn't want to go into that direction. And so the navigation system integrates all of the signals to figure out, basically, on average, how favorable is this direction based on how many nutrients are going up, how many toxins are going up or down, and figures it all out and gets the end calculation, which is how long it will travel in that direction. And so cells, as I've said, cells will swim towards higher concentrations of attractant chemicals like nutrients, and they swim away from high concentrations of repellents, because that's where they want to be. Is they want to be in a place where there's lots of nutrients and not many toxins. So increasing concentrations of attractants or attractant chemicals activate the core signaling units, while increasing concentrations of repellents inhibit the core signaling units. When a core signaling unit is active, it sends out a signal which shortens the run length. So if the concentration of an attractant is increasing, then more core signaling units will be inhibited, which results in less output signal, which then lengthens the length of the run. And then vice versa, if the level of nutrients is going down, then more core signaling units will be activated, which results in more output signal, which will shorten the run length. There's much more to the navigation system than what I've just described here. That's just a basic description, because the navigation system also has to perform calculations to filter out signal noise and also to amplify signals up to 50-fold, which is huge. And there was an evolutionist who was talking about bacterial navigation systems and was saying that bacteria track chemical gradients with exquisite precision, which is true. They're so precise in the way they calculate how far should we go in this direction so that they can navigate in the right direction. Now, it seems that some plants will release specific attractant chemicals into the soil around their roots to attract bacteria to their roots to form a symbiotic relationship with the plant, which benefits both the plant and the bacteria. And so here you've got plants releasing a signal which the bacteria then pick up and swim towards that signal so that they can then get to the root and form the symbiotic relationship. But some bacteria don't just swim towards or away from chemicals, but also swim towards and away from light. So some bacteria perform photosynthesis like plants, and so they need light to make sugar and to get energy. And so these bacteria want to swim towards light so that they can do photosynthesis. But there's other bacteria that swim away from light into dark places, possibly to hide from predators. And so in these species, then they've got light sensors which relay signals to the navigation system, and the navigation system again figures out whether light is going up or down to either swim towards light or away from light. Now, what about the compass needles? Some bacteria navigate using Earth's magnetic field, which is just incredible. I talked earlier about the challenge of figuring out which direction is up or down, because at a small scale, then because of Brownian motion with the jostling, then if your bacterium getting jostled around, then you can't tell which direction is up or down. But some bacteria have a very clever way of figuring out which direction is up or down using Earth's magnetic field. This is because Earth's magnetic field doesn't just travel from the South Pole to the North Pole, but comes up out of the Earth in the Southern Hemisphere and bends back down into the Earth in the Northern Hemisphere. And so by using a compass needle to measure the direction of Earth's magnetic field, a bacterium can figure out which direction is up or down. If it's in the Northern Hemisphere, then downwards direction is towards magnetic north, but if it's in the Southern Hemisphere, then downwards direction is towards magnetic south. If you move one of these bacteria between hemispheres, then at first they move in the wrong direction, but it doesn't take very long for them to calculate that the magnetic field is now in the opposite direction to what it was before. And so they've got some system to calculate which direction the magnetic field is relative to up and down, and they can completely switch the direction so that they go in the right direction. We're not entirely sure how this works yet, but it's pretty amazing that they're able to do that.
That's incredible, David. And from the flagella to the navigation systems, bacteria are amazing single-celled organisms. And this complexity only increases staggeringly when you consider multicellular organisms like human beings. In an article called The Secrets of the Human Cell, Peter Gwynn writes, each of those 100 trillion cells in a human being functions like a walled city. Power plants generates the cell's energy. Factories produce proteins, vital units of chemical commerce. Complex transportation systems guide specific chemicals from point to point within the cell. Sentries monitor the outside world for signs of danger. Disciplined biological armies stand ready to grapple with invaders. A centralized genetic government maintains order. Now, David, can you tell us more about the amazing things in cells?
There's so many amazing things in cells I could talk about. Just every single molecular machine is amazing. I like to think of, the book and movie Horton Hears a Who, about the elephant that discovers that a tiny, or what looks to him to be a tiny speck, is actually a tiny world with a bustling civilization on it. I like to think of cells like that, where most cells are smaller than our eye can see, and yet every one is a tiny little world with this bustling civilization of machines inside them performing all the necessary tasks for life. So you've got machines that manufacture proteins, machines that package cargo into containers, machines that transport cargo between different compartments in the cell, machines that construct three-dimensional road networks in the cell, machines that have mechanical arms and legs to walk along these roads to transport cargo to different parts of the cell. There's machines that construct and assemble other molecular machines, which I briefly discussed earlier with the flagellum. There's machines that deconstruct and recycle molecular machines when they're not needed. Just so many different machines, and it truly is like a civilization inside the cell of machines. I've talked about bacterial flagella motors, but bacterial flagella motors are just one of multiple different molecular machines that bacteria use to move. There's another machine called the pilus machine, which extends long filaments out of the cell, which the tip of them then grab onto a surface, and the pilus machine then pulls the filament back, which then pulls the cell forward across the surface. And the pilus machine can also be used to grab onto other cells and then pull them together, and then they can exchange DNA. There's also another molecular machine, which some species have, which uses, essentially they're like legs, which walk across a surface. And a lot more research is needed to study that, but it's pretty cool to think that there's some bacteria that, in some sense, they walk across a surface with these tiny little molecular machines. There's also another molecular machine which has a rotary motor which drives the rotation of a large helical track, which just like imagine like an army tank with a track, then the rotation of that track then allows the cell to glide across a surface. There's also another species which has a large array of machines which it uses to swim. But again, with this one, we know very little about how these machines work and how they're allowed to swim. So it's another fascinating area of research, which I'm sure there'll be many amazing discoveries in the years to come. There's also bacteria in the ocean that have buoyant gas-filled chambers, and they use these buoyant gas-filled chambers to float up and down in the water column or in the ocean in the same way that man-made submarines have gas chambers to move up and down. Also, with flagella motors, then there are also many other types of motors in bacteria. I mentioned one before in the gliding motility track, but there's also lots of other electric motors in bacteria which power other machines. So there's one electric motor called the Toll QR motor, which during cell division of bacteria, then this motor is recruited to the division site where it then powers other molecular machines which are then involved in the cell division. There's also another motor which, again, it's an electric motor, which powers many different types of transport machines in the outer membrane of the cell. So you've got this rotary motor in the inner membrane, which rotates and powers machines in the outer membrane, which then transport stuff into the cell. And there's so much more that I could talk about with bacteria that make electrical wires over a meter long to power some of their machines, or bacteria that use their whole cell body as a camera eye lens to focus light towards a sensor, which they then use to figure out which direction light is coming from. But I don't have time to discuss those, and there's so many other things I'd love to discuss, but that's just a wee taste of the amazing nanotechnology inside bacteria.
Well, we'd love to have you on again, David, because your insight into the microscopic world is incredible. And so in the last question, you told us about this amazing cell design. And I'm wondering, is it possible to try to implement and copy these designs? I know that the field of biomimetics is flourishing. So can you tell us, what benefit is there to modeling cellular processes according to engineering principles, and how has this revolutionized systems biology?
As I've sort of been showing, every component of cells interact with one another in much larger systems. They don't just work by themselves, but they're part of larger systems. And so systems biology is studying these larger systems and seeing how each of the components in the system interacts with one another to either solve computations or regulate stuff, and just understanding how the much larger system works. And there's been a big revolution in systems biology recently, as you've mentioned, with the realization that biological systems work just the same as man-made systems and computer programs and operate according to the same engineering principles. This has revolutionized systems biology because when we analyze biological systems from an engineering perspective, then we get really useful insights into how the systems work and are also able to make accurate predictions about the bits that we don't quite know yet. With the flagella gene regulatory network, it was originally modeled as working in one way, which for me, from an engineering perspective, didn't really seem to make sense. I thought it would make far more sense from an engineering perspective to work in this other way. And because I believe the flagella gene regulatory network is designed by a master engineer, then I had high confidence that future research would show that the previous models were wrong and that it actually works the way that I thought it should from an engineering perspective. And I was right. Another study has come out which shows that the previous model wasn't correct and that it works exactly how I thought it should. And so this is just one example of many different examples where believing that biological systems are engineered by a master designer leads to really good, accurate predictions about how these systems work. Before I talked about cells as civilizations of machines, but again, they're not chaotic civilizations, but instead, cells are highly ordered with every molecular machine and every process in a cell being controlled by programmed systems. The complexity and sophistication of these systems is just simply mind-blowing. In organisms with multiple cells, such as us humans, then individual cells communicate with one another in even larger systems to control processes throughout the entire body, and also to control things like embryonic development and body clocks. And so there are systems in cells and the body which perform computations without using DNA. A good example is the navigation systems I discussed earlier. Those navigation systems in bacteria solve all of their computations only using protein-protein interactions and interactions with the chemicals and the sensors. However, DNA is central to many systems and cells. I now want to briefly explain how genetic systems work and solve computations, because it's something I didn't really understand a few months ago. But once I did understand it, then it's like, oh, that makes so much more sense. So imagine you've got a gene, and for this gene, then there's an activator protein which activates the gene. Now, when the gene is active, the protein that that gene contains the instructions for gets produced. A messenger RNA transcript, which is called an mRNA transcript, is then made of the active gene, and that transcript is then read by a ribosome, which makes the output protein. So when the activator protein is present, it activates the gene, which then results in the output protein getting produced. So we've got an input signal, which is the activator protein, and an output signal, which is the protein produced from the gene. Now let's say that for this gene, there's also a repressor protein, which turns the gene off. So if both the activator and repressor proteins are present, then the output protein is not produced. So now we've got a computation with two input signals, the activator and repressor proteins, and one output signal, which is the protein that the gene codes for. If only the activator protein is present and the repressor protein is not there, then the output protein will be produced. But if both of them are there, then the output will not be produced. Now let's imagine that this gene is also under epigenetic control. So epigenetics is another system, another code inside cells, which attaches tags to DNA or to proteins that are attached to the DNA. So let's imagine in our example gene that if a epigenetic tag is attached to a specific site in the gene, then that gene will be turned off. So now we've got three input signals controlling the output signal. But now let's say that the mRNA transcript for this gene is also inhibited by another regulatory protein. So if that regulatory protein is there, then the protein will not be produced. So now we've got four input signals controlling the output signal. But now you could add even another layer where most mRNA transcripts in humans are edited by a molecular machine called the spliceosome. This machine cuts or splices sections out of the mRNA transcript and joins the other two bits together. And by cutting different sections of the transcript out, then the protein that gets produced will be different. And so by changing which sections are cut out, you can change which version of the output protein is produced. But how does the spliceosome know which sections to cut out of the mRNA transcript. This is controlled by codes on the transcript itself, but also by the presence or absence of different signals, such as different proteins or regulatory RNAs. So let's say in our example gene, if a particular regulatory RNA is present, then it will cut one section out of the transcript. But if that regulatory RNA is not there, then it will cut a different section out. And so that will change which version of the output protein gets produced. And so now we've got 5 input signals controlling the output signal. But in a cell, many of these gene computations get wired together so that the output signal from one computation becomes one of the input signals for a different computation. And then the output signal from that computation is then the input signal for another computation. And so you end up with what's called a gene regulatory network, where you've got all these different genes all wired together in a network, which functions just like a computer program. And these networks can also have feedback loops, where the output from one gene can activate another gene, which the output from that gene can then go back and inhibit the first gene. And so then you've got a loop structure. And so just like in computer programs, where you've got feedback loops and feed-forward loops, then all of these things are also present in genetic circuits. There's also, what's quite interesting is mechanical signals can also regulate genes. So if a cell gets pushed in a certain way, then pushing the cell in that way can push filaments in the cell, which will result in a section of the DNA changing shape, which will then change the regulation of the genes in that section. Now, in your question, you asked whether humans will be able to copy these designs. Scientists have made some very simple programmed genetic circuits, but they're incredibly simple compared to the circuitry God has designed inside cells. I expect that with time we'll get better and be able to make more complex circuits, but it will take a lot of work from scientists and engineers to be able to copy God's amazing designs.
David, this is incredible information. But there are some who might object to these ideas that you're presenting, and they'll say that, well, the evolutionary processes can adequately account for each of these points that you're raising. For instance, the University of California, Berkeley states, because the central mechanism of intelligent design is untestable, evidence relevant to the idea is lacking. However, some ID proponents have made testable claims that deal more with discrediting evolution than with the mechanism of intelligent design. These claims, for example, that the components of bacteria flagella cannot function independently of one another, have been tested and refuted by the evidence. So, David, is it the case that the evolutionary theory can account for the existence of these molecular machines?
In simple, no, but there's no simple answer. It is true that most proteins in the flagellum, if you remove them, then the flagellum stops working as a propeller. But flagella can and do serve functions other than motility, which is often pointed out by evolutionists. And so flagella are also used to attach to surfaces and to form bacterial communities called biofilms. And they're also used to secrete different proteins and things like that. In fact, there's some non-motile species that don't swim that have flagella and use them entirely for functions other than motility. However, these non-motile flagella are not missing links towards motile flagella, because evolutionists believe that the non-motile flagella evolved from the motile flagella. Also, I remember an ID advocate predicting from his design engineering perspective that the flagellum would function as more than just a swimming machine. This makes sense from an engineering perspective, to have a machine that performs more than just one function. And so, far from disproving design, the discovery that flagella serve functions other than motility actually confirms a design-based prediction. The fact that flagella perform multiple tasks means that there's far greater engineering constraints on the design of the flagellum than if it was just a swimming device, because it means that there's far less designs which, like there could be designs that would work as a swimming device, but they wouldn't be able to perform the other functions. And so the fact that they can do multiple functions just points even stronger to design, because it would be even harder for it to evolve. Now, some flagella proteins can serve functions in other molecular machines. I talked earlier about a secretion machine called the export apparatus, which is used during flagella construction. Recently, it's been discovered that this molecular machine is also used to construct another thing in cells called nanotubes, which are used to transport proteins and other things between bacterial cells. But again, using the same machine to construct two different types of devices is a very smart design from an engineering perspective. This evidence doesn't disprove design. But we do have to be careful with arguments from evolutionists, because they can often be quite deceptive or a bit confusing. For example, evolutionists will say that there's flagella-like proteins which serve other functions in bacteria. What they mean by this is that there are other proteins in cells which share similarities to proteins in the bacterial flagellum, and these other proteins serve other functions in bacteria. But the flagella and non-flagella proteins, though they do have similarities, there are still significant differences. And in fact, the differences are significant enough that the non-flagella proteins could not have evolved into the flagella proteins, as evolutionists claim. I'll explain why with three main points. I'll list the points and then discuss them in more detail. The first point is that functional conversion between proteins typically requires many more mutations than evolutionists assume. The second point is that functional conversions between proteins which require more than two specific mutations are not feasible to have occurred in the evolutionary history of life on Earth, simply due to probability. The third point is that the more proteins which have to functionally convert for a new beneficial function to arise, the worse the problem gets. And it gets worse exponentially with each additional protein. I'll now go through and explain each point in more detail and provide evidence for them. It might get a little technical, but I'll try and explain it as clearly as I can. And for those who want to dive deeper, there will be links in the description. My first point was that functional conversion between proteins typically requires many more mutations than evolutionists assume. Evolutionists commonly use mutational studies to find out how many mutations would be required to convert one protein, which I'll call the source protein, to perform the function of another protein, which I'll call the target protein. In a mutational study, they typically change one amino acid at a time in one of the proteins using a mutation. So they make a mutation which changes the amino acid, and then they test how that mutation affects the function of the protein. They test each amino acid substitution individually to see the effects that each one of them has individually. Typically in these studies, they find that only a handful of amino acids have to be correct for the protein to perform its function. Imagine that for a mutational study performed on the target protein, it is found that only 7 specific amino acids have to be correct for the protein to perform its function. Now imagine that on the source protein, 2 of these 7 amino acids are already correct. Or in other words, they don't have to be changed to match the target protein because they already match it. This then means that there are 5 amino acids on the source protein which will have to be changed to match those on the target protein for the source protein to be able to perform the function of the target protein. it is assumed that if you change those 5 amino acids to be identical to the target protein, then the source protein will be able to perform the function of the target protein. There's a really simple way to test this assumption, and that is to just change those 5 amino acid substitutions and see if the source protein can perform the function of the target protein. And experiments like this have been done, and surprisingly for evolutionists, the altered source protein almost always cannot perform the function of the target protein. Or sometimes it can, but only very weakly.
And when I say weakly, I mean it can be 100,000 times weaker.
So why can the modified source proteins not perform the function of the target proteins? To explain why, I'll have to go back to the mutational studies. In these studies, there's a big assumption made called the assumption of context independence. This assumption is that if a mutation does not affect the function of a protein, then it will never affect the function of the protein, even if combined with other mutations. Or in other words, if you've got 4 mutations, which each individually do not affect the function of the protein, then it's assumed that if all 4 mutations were made at once in combination, then the function of the protein would still not be affected. Even if there was 120 mutations, which when made individually do not affect the function of the protein, it's assumed that if all of them were made at once, the protein would still work just fine. This is a huge assumption because if it's wrong, then it has huge implications on the feasibility of a protein evolving by chance, or protein evolving a new function by chance. And it's somewhat easy to test. Simply make multiple such mutations at once and see if the function of the protein is affected. And again, this has been done. Royal Truman, who's a biblical creationist, empirically showed that this assumption cannot be made by systematically testing the effect of multiple mutations on a protein, which each individually do not affect the function of the protein. He showed that mutations which do not affect the function of the protein when made individually do affect the function of the protein when made in combination. He found that the more such mutations that were made at once, the less likely it was that the protein will work. And so by the time you get to just 4 mutations, then if all 4 mutations are made in combination, then there's almost no possible combination of 4 mutations which will still result in a functioning protein. And this has huge implications for the evolution, because it means that far more amino acids have to be correct for the protein to work. This also means then that for a source protein to functionally convert to a target protein, it will require far, far more mutations than just the 5 or so predicted using the assumption of context independence. And Royal Truman explains the implications of this, saying that the chance of a functional protein arising by chance becomes, quote, overwhelmingly improbable. Now, this result is logical. Imagine if we had the function of a protein that its function is to bind to another protein. That protein will have a binding site made of multiple amino acids, which will each form a chemical bond with the other protein. Let's say that this binding site has 30 amino acids that each form a chemical bond with the other protein. If a mutation changes one of those amino acids in a way that stops that bond forming, then it won't really have an effect on the protein function, because there will still be 29 other chemical bonds that are still bonded to the other protein. And in fact, for any one of those 30 bonds, if you mutated them individually, then the function of the protein will not be affected. However, if all of these mutations were made at once, then there would be no chemical bonds to bond with the other protein, and the protein would be completely non-functional. Even if half of the mutations were made at once, then the protein might not be able to function properly because the binding site would be a lot weaker and it wouldn't be able to bind strong enough to the other protein. It makes sense that mutations that do not affect the function of a protein when made individually would affect the function of the protein when made in combination. So, my first point is that functional conversion between proteins almost always requires many more mutations than evolutionists assume. My second point is that functional conversions between proteins which require more than 2 mutations are not feasible to have occurred in the evolutionary history of life on Earth, simply due to probability. Douglas Axe, who's an ID advocate, made a computer model to see what is the largest gap that could be crossed between 2 proteins with different functions. In other words, if a functional conversion requires 5 specific mutations to all exist at the same time in the same cell, how long on average would it take for that functional conversion to occur purely due to random chance and random mutations? He made 2 models. In one model, then the mutational intermediates between the source protein and the target protein were neutral to the survival of the cell, whereas in the other model, the mutational intermediates were harmful. If the mutational intermediates were neutral, he showed that the largest functional conversion that is likely to have occurred in the evolutionary age of life on Earth, which is 3.5 billion years, he showed that the largest gap which could be crossed in that time was one that required no more than 7 mutations. Any functional conversion requiring 8 or more mutations is overwhelmingly improbable to have occurred in 3.5 billion years, simply due to random mutation. Now, in the second model he made, where the mutational intermediates were harmful to the cell, the largest gap that is feasible to have been crossed in 3.5 billion years is just 2 mutations. So, which of these models is more likely to be correct? It's well known that producing non-functional intermediates or non-functional proteins wastes a lot of energy for the cell, and so non-functional intermediates are harmful to the cell, and this is well known. What this means is that the second model is more correct, which means that 2 mutations is the largest gap feasible to be crossed by evolution. He also changed the model to see if he grossly favored evolution, would there be more mutations possible? And yet, even in the model where grossly favored evolution, the gap only changed from 2 to 3 mutations. So it doesn't really help evolution that much. Now, this has huge implications for the evolution of new protein functions. It means that all of the functional conversions I discussed earlier that require dozens of mutations, or even if they require just 5 mutations, then they're virtually impossible to have occurred in the evolutionary history of life on Earth. In all of the studies I mentioned above, the source protein and the target protein were very similar to each other, and yet they still require dozens of mutations to convert from one to the other, which means that all of these conversions are virtually impossible to have occurred by evolution. Now, if we consider something like non-flagellar proteins becoming flagellar proteins, then it gets a whole lot worse, because the differences between non-flagellar proteins and flagellar proteins are far more than the differences in those source and target proteins discussed earlier, which means that there's far more mutations which would be required, which means that the chances of those gaps being crossed is just the average time it would take is well beyond the evolutionary age of the entire universe. So to believe that such a conversion could have occurred is basically to believe a naturalistic miracle. My third point is that the problem is multiplied with each additional protein functional conversion which has to occur for a new beneficial function to arise. And the problem gets worse exponentially with each additional protein required. So there are many functions in cells that require multiple different proteins. And in some of these cases, if you remove one of these proteins, then the rest can't perform the function of that system. And so for functions like this to evolve, or for systems like this to evolve, then multiple different proteins would have to functionally convert before any benefit is provided to the cell. And so when someone considers something like the bacterial flagellum and its associated systems, which have over 50 proteins, then, and like each of those have binding sites to other proteins, then the chance of something like that evolving, it's crazy to claim that that was possible because, like, as I've shown, it just couldn't happen. Now, some evolutionists might argue that the flagellum could have assembled via a series of intermediates, with each intermediate having more proteins than the one before, and each intermediate performing a different function, such as a secretion system or an adhesion device. But even if the functional conversions from one intermediate to the next required 3 new proteins to functionally convert, just one of those jumps between those 2 intermediates would still be far beyond what is possible via evolution.
Exactly. And David, we've actually now arrived at our final question. So in light of all of this, how has your research grown your awe of God?
My research into this has grown my awe of God so much. Like, there has actually been times where I've been close to tears just as I've just sat back and reflected on the effort God has gone to and the detail he has gone to in designing every single little molecular machine and system inside cells. And it all just brings so much glory to God. And then to think about how many different types of molecular machines are inside a single cell, and then how many different types of cells are in multicellular organisms, and then how many different types of organisms there are on Earth, it just blows my mind. And it appears that every species on Earth probably has a collection of molecular machines and nanotechnology that's not used by any other species. And so when you consider how many different species are on Earth, then you can begin to grasp just how many different types of molecular machines and nanotechnology there are in living things. And we've only studied a very small handful of the species out there in depth. And so there's still so much more to discover. And I also think, if God puts so much effort into programming the growth of a flower, which is here today and gone tomorrow, then how much more does he care about us?
Amen, David. And thank you very much for your time. It was an absolute joy. And to our listeners, thank you very much for taking the time to learn with us on Current Topics in Science, where scientific discoveries are examined in light of the origins issue. You can find David's biographical information and his articles on the E. coli electric motor in the description, along with the other items that he mentioned during the interview. Please share and subscribe to the Current Topics in Science podcast. It's available on iTunes, Stitcher, Google Podcast, Spotify, and iHeartRadio. Thanks again for listening, and remember, the truth saves.